Muy buenas tardes, bienvenidas y bienvenidos a este acto humilde desde el punto de vista de la universidad, pero de un gran valor para nosotros por poder tener a Robert Langer con motivo del premio BBVA Fronteras del Conocimiento que acaba de recoger ayer mismo en Bilbao. Y es una satisfacción enorme que haya podido agendar un hueco en su apretado viaje para poder estar con nosotros aquí en Santiago y aquí en nuestra universidad. Agradecemos la presencia de la profesora María José Alonso, que es la que ha impulsado la candidatura de Robert Langer para el premio mencionado, y que mantiene pues, ella y otros investigadores una estrecha conexión con él. Agradecemos también, está también la secretaria general de la universidad, y agradecemos muy especialmente la presencia del vicepresidente primero y consejero de Economía, Industria e Innovación de la Xunta de Galicia, don Francisco Conde. Estamos a la espera también de que se incorpore el consejero de Sanidad de la Xunta de Galicia, cosa que también agradecemos. Y, por supuesto, la de todos ustedes, los rectores Barro y Viaño, vicerrector, decanas, decanos, investigadores, directores de centros, profesoras, profesores. Un placer poder contar con su presencia en el día de hoy. Va a tomar la palabra, en primer lugar, la profesora María José Alonso Fernández. Tiene la palabra.
Magnífico señor rector, secretaria xeral, vicerrectores, señor vicepresidente de la Xunta de Galicia, autoridades académicas y políticas y sociales, compañeras y compañeros. Muchas gracias por estar aquí en este día tan importante para la USC. Voy a decir unas palabras en galego, aunque no me defiendo muy bien, y después diré unas palabras en inglés dirigidas al profesor Langer. Tampoco me defiendo tan bien. Y luego, finalmente, hablaré en español. Hoxe é un día grande para a nosa universidade e é un día moi importante para min como persona e como profesional da USC. Síntome moi honrada de que a nosa universidade teña ben conceder a Insignia de Ouro desta universidade ao profesor Robert Langer, a persona que tivo e ten unha influenza extraordinaria na miña carreira, polo moito que me inspira e polo moito que me apoia. E síntome tamén moi satisfeita, xa que estou convencida de que a nosa universidade non atopará un mellor embaixador. Voulle dedicar unas palabras ao profesor Langer e farei unha presentación en castelán a continuación. Dear Professor Langer, dear Bob, today is a remarkable day for our university. It is an unforgettable day for me. I have been dreaming about bringing you to my hometown, to my university, since I was a postdoctoral visiting student in your laboratory 30 years back. I can tell you that when I left your lab with tears on my face in 1992, I told myself, I want to be in touch with this extraordinary person. And the truth is that since then, I have always thought of you as my mentor. Thank you, Bob. I was extremely happy when you received the Príncipe de Asturias Award in Oviedo, and also when you became a member of the Royal National Academy of Pharmacy and came to Madrid. But today, my dream has come true. Today, we are in this historical building, the Fonseca Palace, the first and original building of the University of Santiago de Compostela, and you are receiving a special recognition, the Gold Insignia of the university. Thank you very much for accepting this honor, which is a symbolic expression of our gratitude to you. By being my mentor, as well as the mentor of several of my fellows who are here today, you have not only impacted our lives, but also the reputation of the USC. I also want to express my gratitude to the rector and to my university for supporting your nomination for the Frontiers of Knowledge Award that you just received and for approving this special recognition. Algo que a lo largo de mi carrera científica he apreciado enormemente es la creación de vínculos de amistad y de afecto que nos unen a las personas que colaboramos y compartimos no solo nuestras ideas y hallazgos, sino también nuestras experiencias personales. En mi caso, Robert Langer ocupa un lugar muy especial en este círculo de afectos y he de reconocer que de este círculo forman parte también muchos de sus discípulos a quienes tuve el gusto de conocer durante mis múltiples estancias en el Instituto Tecnológico de Massachusetts. Con muchos de ellos que han ya visitado esta universidad, mantengo una relación científica y personal que valoro muy especialmente. Y de este círculo forman parte también varios de mis discípulos que el profesor Langer ha acogido muy amablemente en su laboratorio. Por todo ello, me siento muy agradecida. Durante mi primera estancia postdoctoral en el laboratorio del profesor Langer, en 1991-92, aprendí lo que era la investigación translacional, el trabajo interdisciplinar y la inteligencia colectiva. Pero, sobre todo, aprendí a valorar mi propio trabajo. Y esto fue gracias a la enorme confianza que el profesor Langer depositó en mí. Nunca olvidaré que a los 2 meses de llegar a su laboratorio, me pidió que lo representase en una reunión que tenía lugar en la Organización Mundial de la Salud, en Ginebra. Y eso fue solo un pequeño ejemplo de su confianza. Uno de los aspectos más distintivos de la personalidad de Robert Langer es su apuesta por la atracción y desarrollo de talento hasta la más alta expresión. Contagia su entusiasmo por la ciencia y convence a todos los que le rodean del valor de su trabajo y de su impacto en la sociedad. Para mí, su influencia supuso un cambio radical en mi forma de afrontar la ciencia. Después de esta experiencia inicial, a lo largo de estas décadas, he realizado numerosas visitas a su laboratorio en las que he disfrutado de su total disposición a acogerme y a compartir conmigo sus notables avances científicos. De hecho, este mes de julio estaré nuevamente en su laboratorio. Creo que la mayoría de los presentes sabemos que el profesor Langer es el científico más sobresaliente en el mundo en el ámbito de los biomateriales y de su aplicación en biomedicina, notablemente en el campo de la nanomedicina y la ingeniería de tejidos. Los datos que se reflejan en su currículum lo dejan muy claro. Nadie lo ha igualado, ni desde la óptica del descubrimiento, ni desde la perspectiva de su traslación a la práctica clínica. Nadie como él ha podido conjugar la ciencia al más alto nivel, reflejada en más de 1000 publicaciones científicas de alto impacto, con la creación de decenas, ayer comentaba 40 empresas, que han permitido la explotación clínica de sus hallazgos. De hecho, él ha concebido y creado diversas áreas de conocimiento. Por ejemplo, él fue el primero en desarrollar, en los años 70, nos hablará en su intervención, los primeros sistemas de liberación controlada de biomoléculas. Él fue quien desarrolló el concepto de vacunación monodosis, es decir, una sola administración, un concepto que seguidamente adoptó la Organización Mundial de la Salud y en el que yo me vi involucrada, pude participar durante mi estancia postdoctoral. Seguidamente, a mi vuelta ya a la Universidad de Santiago, pudimos colaborar en el desarrollo de vacunas no inyectables, vacunas nasales, gracias a la financiación de la Fundación Bill y Melinda Gates. Otro de los grandes hallazgos de su laboratorio, en el que también tuve el gusto de involucrarme, es el de la nanotecnología aplicada a la liberación de ácidos nucleicos, es decir, RNA, en particular RNA mensajero. Recuerdo el momento en el que la terapia génica y las vacunas RNA alcanzaron su punto álgido y, como suele ocurrir con la ciencia disruptiva, ese punto de extrema atención vino seguido de una fase de desinterés, hasta la década de los 2000, desinterés que fue motivado por muchos detractores, que eran esas personas que esperan que la ciencia tenga resultados inmediatos, y nunca es así. Curiosamente, recuerdo una conversación que tuve con el profesor Langer en ese momento en el que parecía que las terapias RNA eran una ilusión más que una realidad, hablamos de hace 15 años, y en esa conversación él me habló de la creación de la compañía Moderna, que por cierto nos suena muy bien en español, pero lo que significa en realidad es mRNA, Moderna. Y de su conocimiento de que él me dijo en aquel momento de que estaba convencido de que estas tecnologías tendrían un gran impacto en la medicina, a pesar de los muchos detractores. ¿Cómo pudo predecir el profesor Langer que estas vacunas de RNA mensajero iban a salvarnos de una gran epidemia? Su lema ha estado siempre claro: cuando el talento y la financiación se unen, se consiguen resultados extraordinarios. La apuesta por el talento y por la economía de la empresa Moderna fue verdaderamente excepcional y su fruto lo ha sido aún en mayor medida. Al día de hoy, todas las grandes farmas están haciendo uso de la nanotecnología que permite la liberación de material genético para desarrollar no solo nuevas vacunas, sino también terapias personalizadas y terapias avanzadas. Por todo ello, no es sorprendente que la Fundación BBVA haya reconocido al profesor Langer con el Premio de Fronteras del Conocimiento en Biología y Medicina por su excepcional contribución al desarrollo de las modernas vacunas que nos han permitido combatir la pandemia COVID. Creo que hoy más que nunca debemos sentirnos muy agradecidos al profesor Langer. Podemos decir sin ningún género de dudas que sus contribuciones han salvado miles de millones de vidas, han salvado nuestras vidas. Estos son solo algunos ejemplos de sus brillantes ideas y desarrollos que han cambiado el mundo. No es de extrañar, por tanto, que su trabajo haya sido reconocido con cientos de premios, algunos de ellos equivalentes a lo que podría considerarse el Premio Nobel en el ámbito de la bioingeniería, y otros particularmente emblemáticos, como es el Premio Príncipe de Asturias y el Premio Queen Elizabeth en Inglaterra. Quiero señalar también que fue el primer ingeniero en formar parte de 5 academias en Estados Unidos y al día de hoy forma parte de decenas de academias en todo el mundo, entre otras la Real Academia Nacional de Farmacia. En fin, creo que no debo consumir más tiempo en describir sus honores, están visibles en las redes sociales y en la web. Prefiero referirme a la noción que él tiene del impacto de su trabajo. Como él acostumbra decir, el impacto no reside únicamente en sus publicaciones científicas, ni siquiera en sus empresas, sino en las más de 1000 personas que se han formado con él y en los millones de personas a quienes cada día inspira con su trabajo. Creo que, como ha sido definido ya, el profesor Robert Langer pasará a la historia como uno de los grandes héroes que ha cambiado nuestras vidas. Dear Bob, my special thanks for your continuous inspiration, advice and support all along my career. Thank you as well for your support to my fellows. Thank you for your contribution to saving our lives. And thank you for dedicating this precious day to all of us. It is a great honor for us to have you receiving the gold insignia of the University of Santiago de Compostela. Thank you.
en uso das atribucións que me confire o ordenamento xurídico, de conformidade co establecido no artigo 162.2 dos Estatutos da Universidade de Santiago de Compostela e no Regulamento de Honras e Distincións da Universidade aprobado polo Consello de Goberno o día 14 de marzo de 2008, resolvo conceder a insignia de ouro da Universidade de Santiago de Compostela ao doutor Robert Langer, profesor do Instituto Tecnolóxico de Massachusetts, pola súa relevante traxectoria docente, investigadora e de transferencia no ámbito da biomedicina e a súa estreita colaboración coa Universidade de Santiago de Compostela. Santiago de Compostela, 16 de xuño de 2022, o reitor Antonio López Díaz.
This is the one. Oh, okay, that'll be good. Yeah, perfect.
Thank you so much. It's an honor for me to be here today, and Maria, thank you for that touching speech. I feel very honored to be here. I have had the good fortune of having a number of connections with Spain, and particularly with this University of Santiago. You know, 4 people from here, Maria Alonso, Maria Tobio, Alejandro Sanchez, and Cecilia Praga, have all been in our laboratory working, and all did an unbelievably great job. We also had a collaboration on, have one on vaccine protection, and finally, I'm just very grateful about the BBVA nomination, and was fortunate enough to receive it, and Maria was there last night. You know, Maria asked me if I would, Professor Alonso asked me if I would tell you a little bit about my life and career. So I'll do that. You know, I'm a chemical engineer. I got my chemical engineering degree in 1974. And when I did, pretty much all my colleagues went to the oil industry. And I wasn't that excited about it. They were talking about just increasing the yield of oil by tiny percentages, and yet that would be worth enormous amounts of money. But to me, it seemed kind of boring. So I started to think, how could I use my chemical engineering background to help people? One of the things I thought about was education of high school students even, and developing new curriculum. When I was a graduate student at MIT, when I was supposed to be doing my thesis, I helped start a school for disadvantaged high school students in Cambridge, Massachusetts. And I got very involved in developing new chemistry and math curricula. One day I saw an ad to do just that at what's called City College of New York. So I wrote them a letter applying for a job, but they didn't write me back. But I really liked the idea, so I found all the ads I could to do that. I found about 40 of them, and I wrote them, and actually none of them wrote me back. So I wasn't doing so well. So I thought, well, what other way might I use my chemical engineering education to help people? I thought about medicine. So I wrote to a lot of hospitals and medical schools, and they didn't write me back either. One day I was walking into the lab, and one of the older scientists said to me, he said, Bob, he said, there's a surgeon in Boston named Judah Folkman. And he said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. Now, Dr. Folkman had this idea, and this is on the first slide. And his idea, this is from the New York Times in 1971, but his idea was that if you could stop blood vessels from growing, that might be a whole new way to treat cancer. When you look at this picture, what you see is, if you follow it, the tumor starts out, it's abnormal, it grows as like a 3-dimensional mass, And when you get to the middle bottom of the screen, what you see is that maybe it's 1 million cells. And then it turns out, he said, it runs into a nutrition problem. Cells in the center die because they can't get nutrients. What he said, though, is he had a theory that the tumor made a chemical signal. He called it TAF, or tumor angiogenesis factor. And that would cause the blood vessels, which normally don't do anything, to start growing, as you can see in the bottom of the screen, to the tumor. And when that happens, then that solves the nutrition problem. The tumor gets bigger and bigger. And when you go to the upper right, you see it very vascularized. And it can actually spread through those blood vessels. That's a process called metastasis. That often is what kills people. His theory was that if you could stop blood vessels, maybe that would be a whole new way, achieve anti-angiogenesis, a whole new way to treat cancer. So it turned out that this theory, almost everyone at that time criticized him and thought it was wrong. I knew nothing about biology. I thought it was great. So he offered me a job. It was the lowest paying job I got, but I thought this was a way you could do something really important. It turns out when I joined his lab, I was the only engineer in the entire hospital. And he asked me to do 2 things. One, he said, well, I want you to prove my theory. And 2, in so doing, I want you to isolate the first substances that could stop blood vessels from growing. One of the key things in doing this, whenever you have a new biologic phenomenon, is having what we call a bioassay, a way to study it. And there wasn't any. So we had to invent one. And our idea was that, and it's not easy to do because there's always background blood vessels in almost every organism, every part of the body. But one place there isn't is your cornea. And so our thought is there's certain types of tumors you could put in the eye, like what's called the V2 carcinoma, that will cause blood vessels to grow. And then we needed a tiny little delivery system, like a micro or nanoparticle that we could put right next to it that could deliver the different blood vessel inhibiting molecules. And it turned out all the ones we were isolating were large molecules, macromolecules. So no one had ever developed tiny particles to deliver macromolecules before. In fact, there was only one company in the world at that time working on this idea of drug delivery. It was a company called Alza in California, and Dr. Folkman was on their advisory board. And they had many famous people on that board, including Nobel Prize winners in chemistry and so forth. And he asked them, could they help us? But all of them came back and said that they could not help us. They basically said that you could never do this because these large molecules they're just too big to slowly diffuse through solid materials. It's like none of us, we would argue, could walk through a wall like that. We're just too big. But if we were going to solve this blood vessel problem, we had to solve the delivery problem. So I spent 2 years working in the laboratory, experimenting with different techniques, and I actually found over 200 different ways to get this to not work. But eventually, I found one way that did. And I was able to figure out the different materials, different solvents, different procedures, and I was able to make little micro and nanoparticles. You see a picture of one shown here on the left and cut in half on the right. And we showed in the British journal Nature that you could use this approach to literally deliver any large molecule, including, this is the first report, that you could use tiny particles to deliver nucleic acids. Nucleic acids are like DNA and RNA. So let me go back 3 months before this paper came out in Nature. I got asked for the first time in my life to give a major talk at a big scientific meeting. I was incredibly nervous. I was 27 years old. It's 1976. I practiced this talk for weeks. I finally got up and I gave this talk. And I thought at the end of it, I didn't do that badly. I didn't forget too much what I was going to say or stammer too much. And I thought when I was done with this talk, all these older scientists, being nice people, would want to encourage me, this young guy. But when I got done, I stepped off the podium, and a whole bunch of them came up to me and they said, "We don't believe anything you just said." They said what you heard before, that the large molecules were too big to diffuse through solid materials. Also, the way we were making the different particles, we were using organic solvents like ethanol and methylene chloride. And they said that would destroy anything we put in that was like really big, like a peptide or protein or nucleic acid. And then actually things went downhill from there for me, if that were possible. You know, I tried to get research grants to support my work. And I would write research grants to the government, what's called the National Institutes of Health, and they have reviewers who review them. And my first 9 grants were rejected. In fact, I still remember one time I got 2 grants rejected the same day. One of the grants came back, it was a cancer research grant, and the reviewers, it was a pathology study section, and they said, well, Dr. Langer should not receive this. He's an engineer. He knows nothing about biology, and even less about oncology. Now, even I thought I knew enough math to know that wasn't possible, but it didn't really matter. I didn't get those grants. Also, I really liked being a postdoc, but a lot of my friends said it's probably not good to be a postdoc forever. So I started applying for faculty positions to chemical engineering departments, because that's what I was. No chemical engineering department in the world would hire me. They said this chemical engineer should be doing oil and things like that, not biology. So I didn't get any jobs. But Dr. Folkman knew another person on that ALSA advisory board, a man named Nevin Scrimshaw. And he introduced me to him. And Dr. Scrimshaw, he was the kind of department head who was kind of like a benevolent dictator. So he liked me. He offered me the job as an assistant professor, but he didn't bother to ask anybody else in the department what they thought. Now that might have been okay, except for one thing, which is that the year after I joined the department, Dr. Scrimshaw left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. In fact, Michael Marletta is a good friend of mine, and he was on the faculty about the same time I was. And a number of years ago, when I got an award in the US, they asked him to make some comments. So I excerpted this one. It was actually published. And he's talking about what it was like to be with me at MIT in the late 1970s. And I'll just read this to you. He said, one evening I went to a faculty dinner at a Chinese restaurant with myself and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. So that I had a pretty inauspicious beginning. But I felt that the work we were doing, I could see that the drug delivery systems, at least I felt, were working. So I felt, well, we ought to be able to now maybe solve the angiogenesis problem. I had, in the meantime, isolated maybe 100 different fractions, and we had these bioassays, so we could test it and see, could we ever stop blood vessels? No one had done that before. So we literally tested maybe with surgeons 2,000 rabbit eyes. We maybe would do an N of 20 for each fraction. Pretty much every fraction we tested did not work. I always think of those as like the controls. But one did. And I apologize for showing you what I'm going to show you in advance, but there's no other way to illustrate this. I'm going to show you rabbit eyes. They're very bloody, but you'll see the difference. So don't look if you don't like blood, but here it is. And this is from a paper we published in Science in 1976, the same year as we published that Nature paper. It was the first demonstration that blood vessel inhibitors did exist. And what you see, and on the left-hand side, is what normally happens. A sheet of blood vessels grows over the delivery system towards the tumor. A few weeks later, if you allowed this to continue, the tumor would be three-dimensional, grow out of the orbit of the eye. It would actually kill. We sacrificed the animals before that. And this happened every single time, probably out of 2,000 eyes. But when we put this one inhibitor in, it was a fraction, it wasn't totally purified, what we saw was striking. What you see on the right, and we saw this 20 times, and we can even quantitate it by the length of the longest vessel. But what you see is the vessels are lower, they're sparser, and they avoid the tumor. 60% of the time, these tumors never grew. So this proved for the first time that these blood vessel inhibitors did exist, and it would provide a way that all future inhibitors could be tried isolated to be studied. Now, as an aside, it took 28 years from this paper we wrote in Science before the first blood vessel inhibitor would get approved by regulatory authorities such as the FDA. It cost billions of dollars and work by companies like Genentech and others before this would happen. But starting in 2004, 28 years after this paper, something startling happened. One, and this may be hard to see in the back, but what I want to illustrate, and this is not even a complete list, but what you see is one after the next blood vessel inhibitor approved by the FDA. Avastin, the first of these in 2004, is probably the #2 best-selling biotechnology drug in history. And not only are these used for all kinds of cancers, but drugs like Eylea and Lucentis provided for the first time ways to treat disease of blindness in the back of the eye, which also is caused by abnormal vascularization. They are also blockbuster drugs. And these, like I say, have been used by many millions of patients every year. Dr. Folkman also said to me one day, he said, Bob, we should file a patent. You know, that, for those of you who follow my career, that might not seem so surprising. But you know, in 1976, I had 0 patents. And Boston Children's Hospital, which was one of the best hospitals in the world for children, they had 0 patents too. So we filed for a patent for the first time. And 5 years in a row, the patent examiner rejected it. And I remember the head of the hospital and technology transfer office came to see me one day, and they said, Bob, you are wasting so much money on this patent fight. They said, just give up. They're never going to allow it. But I don't like to give up, as you can probably tell by now. So I started to think, how could we convince the patent examiner to allow this patent? Legally, of course. And science really wasn't working. But I mentioned to you in the beginning, when we first did this, all these people told us, Dr. Folkman and I, that this was impossible. It could never work. I wondered, maybe one of them wrote that down somewhere. So you can do what's called the science citation search. And I did this in 1982, 6 years after that Nature paper came out, to see who cited it and what they wrote. And I actually found it's very fascinating to see what people have to say about you. So I did this search, and I found a number of interesting articles, but let me just show you a quote from one of them. And this was from 5 of the top material scientists in the world. And what they wrote, describing this very young field of drug delivery, is they said, generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising is a great word for a patent examiner, some surprising results that clearly demonstrate the opposite. So I showed this to the people at the hospital, and they flew down to Washington to show it to the examiner, and he said, gee, I had no idea. I'll tell you what, I will allow this patent if Dr. Langer can get written affidavits from each of these 5 people that they really wrote this. So I wrote them, and they were all nice enough to write me back that they really wrote it. And as a consequence, that patent got allowed. It was a very, very broad patent. And with that, you know, it's the first patent on delivery of macromolecules. And with that, we got involved in trying to teach companies how to do this. I got involved in starting companies. Today, based on this, and a lot of those companies my students work at, there's all kinds of products that are approved by FDA. I just put a few of them here, but if somebody has advanced prostate cancer or endometriosis, there's a drug, luteinizing hormone, releasing hormone analog, and you can put it in little microspheres, and that will protect it from being destroyed. You can't swallow it, it's just too big to be absorbed. If you inject it, it works, but it's destroyed in seconds. So you have to put it in these little micro or nanospheres, otherwise it won't last long enough. And there's all kinds of these different ones. I need to go back. Yeah, there's all kinds of different ones on there. Drug-eluting stents, if anybody has heart disease, that's on the bottom right. But all these are kind of implants. And they're all kind of implants or microspheres that you might inject underneath the skin. Now, we'd also, even going back to that Nature paper, use those approaches we developed to make nanoparticles, too, which you could inject into the bloodstream or muscle or whatever you wanted. But when you made nanoparticles, you had additional problems. So the surface area is so great, sometimes they stick together, and also there's cells called macrophages that eat them. So what we did is we came up with a disguise. Ruxandra Graff and others in our lab, Vladimir Torchelin worked with us, and we came up with a disguise by adding polyethylene glycol, an FDA-approved material, to the surface. And then water comes to surround it, And our thought is that disguise might work because cells normally see water, so maybe they won't eat, these nanoparticles. So there's a TV show in the United States called Nova, and they once had a show called Making Things Smaller. And they came and spent quite a bit of time filming in our lab, and they made a video, which I thought I'd show you because it explains this far, far better than I do. I hope. Let me just go back. Is there something I'm supposed to press to get the video working? Do you know?
He starts with a nanoparticle
Yes, I
of
did it.
anti-cancer drug. That gets encased in a plastic that releases the drug over time. That, in turn, gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
I should point out to you that the clinicians I work with tell me it doesn't really blow the cells up quite like that. But you get the idea. And the thought then is that you could use, like I say, we published this in Science, and the thought was that, that we could use these particles to deliver small molecules like you see, but even more exciting might be the possibility of someday using this to deliver genetic therapies like small interfering RNAs or messenger RNA. And with respect to the latter, and Maria pointed this out in her introduction, we've started a number of companies. I've been involved in advising a number of companies. But in 2010, Derek Rossi, Nubar Fayan, and Ken Chen and I had the idea that we would start a company to create therapeutics and vaccines based on messenger RNA. I mean, the idea really goes back, to thinking about how successful have protein therapeutics been. And it's amazing. When I look back to the 1970s, when I started out, there were almost no protein therapeutics, maybe just insulin. But if you look at the last 20 years, 70% of the top-selling drugs are proteins. It's pretty remarkable. But proteins, as you may know, take a long time to make. You have to grow up eggs. It may take years. You have to sometimes have giant cellular factories. And it takes a long time, so you might have to guess at how well you're going to treat the disease, like, say, flu, because you don't know exactly the right thing to make. Also, there's a lot of diseases that you can't necessarily treat with proteins, diseases that are intracellular or membrane-bound. So our thought was that, what if we made messenger RNA, put it in nanoparticles, which you have to do to protect it, then could you make drugs and make vaccines? So we started this company, like I say, in 2010, and we were kind of rolling along. I would say probably for the first 10 years, lots and lots of people criticized the company. They said this idea would never work. Nonetheless, we were still, by the early 2020, when COVID hit, we were in 13 clinical trials, 8 with vaccines. There was nothing approved, but I remember all kinds of people calling up and saying this was a ridiculous idea, the stock was way overvalued, and things like that. So, like I say, so what happens is January 2020 comes, and we recognize, Stefan Boncello, our CEO, and others, we recognized that this could be a problem. So we started to make the COVID vaccine. Let me just show you the timeline, which I think is really historic. The Chinese scientists published the virus genetic sequence on January 11th, 2020. It took less than 2 days for the Moderna scientists to design the vaccine, then put it in nanoparticles, make batches, and ship them to our clinical partner, the National Institutes of Health, on February 24th. And the first patient was treated just a little less than a month after that, on March 16th. So what you do when you're doing clinical trials, the first trial is usually aimed at safety and finding the highest effective dose. So we did this, and that started in March, but by May 19th, by May 19th, a little over 2 months later, we had the first data, and we announced that data. So you'd like to think, given the problem and how serious it had become by then that people would be cheering us on. Nonetheless, almost everybody in the United States criticized us. They said, well, I'll just show you. The Boston Globe, my local newspaper, had talked to a lot of scientists. And here's what this is May 19th, 2020. So their front page headlines is that this is not how you do science. And you can see whose picture's underneath it, which is me, which was very disappointing that we'd get this kind of reception. But I want to go 6 months later. 6 months later, it's November. And now we've not only done the phase 1 trial, we've done phase 2 trials and what are called the phase 3 trials, which is really the large trial that is supposed to tell you exactly what the regulatory authorities need. We did 30,000 patients, 15,000 controls, 15,000 treated. And I remember being in the boardroom in November when we broke the code, and it was striking. Of the treated patients, of those 15,000, not a single one died, not a single one went to the hospital, and it was 94 to 95% effective. For reference, flu is maybe 45% effective. There was one other system that also worked, also putting messenger RNA in nanoparticles, which was BioNTech, and they were working with Pfizer. But to me, it was a giant victory for science, a giant victory for nanoparticle technology. And what would happen is not only would this get approved for emergency use authorization and later fully approved, it's now treated over 3 billion people around the world. Let me just show you some data, since sometimes people still don't think it works. Here's just quick some data. This is weekly hospitalizations in the United States. By the way, from everything I hear, Spain is doing a lot better in terms of, not being so ridiculous as some of the people in our country, but and thinking it doesn't work. But basically, here's data, and you see if you look at hospitalizations per 100,000, you see the huge difference between unvaccinated, the orange bar on the top, vaccinated with no booster, and vaccinated with a booster. By the way, all the data I'm showing you now is 2022 data this year. Let me also show you data from the Mayo Clinic, showing you that Moderna is 94% effective, BioNTech 91% effective, Johnson & Johnson, which is, these are the 2 mRNA vaccines and nanoparticles. There are other vaccines, but none of them have become close to that being nearly as effective. The Johnson & Johnson one, which is an adenoviral vaccine, was 66% effective, but had some side effects, so it was taken off the market. Sort of standard protein vaccines made by GlaxoSmithKline and Sanofi were about 57%. There's ones made by different Chinese companies too, but all were far less effective. Also, just to take this further, if you look at publications that really examine this in detail, including some of the most tightly reviewed publications in the world, like the New England Journal of Medicine, which probably has an impact factor of 100, probably the most widely cited journal ever. Here, for example, you can see almost 400,000 people, this was a study in Qatar, getting the 2 best vaccines, the one from Moderna, which is mRNA 1273, and the BioNTech one. And you can see that the lowest incidence of infection was with the Moderna one, with those 4, and it was incredibly low. And the last slide that I'm going to show you on this is from the U.S. This is U.S. Army veterans, and here there's over 400,000 patients. And now we're looking at hospitalizations, and you see it's also incredibly low, like even after half a year pretty much, it's like 0.05% incidence of hospitalization, incredibly small. When they have these vaccines. So there's still more to do. Moderna is now in something like 25 clinical trials, including having announced a week or 2 ago of very successful 8-fold improvement by using the same technology I just showed you to make a vaccine against the Omicron variant. And many other clinical trials, too, for flu vaccines, combination vaccines, heart disease, and so forth. So it's my hope that we'll not only see treatments for COVID that have come out of this, but treatments for many, many diseases that have never been able to be treated before. Now, that doesn't stop us from going back to our MIT lab and still trying to change the future. And I'm going to now go back to 2010, or 2011. And one of the big things that you may know is issues that come up is trying to help people in the developing world. And I've always been interested in not only creating new technology, but trying to reach as many people as we can. That's why Maria went to the World Health Organization back in 1992. But in 2011, I had a visitor. And this visitor was a software entrepreneur, but he was very concerned about public health. And he was interested, and he read that we'd solved a number of different problems to help different people, and he asked, could we help people in the developing world? Let me just show you a picture of me and him. That's me on the, there, and if you don't recognize him, that's Bill Gates. That's my office. And they've given us probably close between our lab and companies we've started to help on this, about $200 million to try to, over the last 10 years, to try to really advance many different things. And we're now in 9 clinical trials just from things that have done in our lab to help people in the developing world. Everything from new nutrition to pills that can last for super long times. But I want to, again, just pick an example to you, which is vaccines. And one of the things that he and his colleagues were interested in, as you may know, people in the developing world, they don't come back very often for a second injection. By the way, a lot of times people in the United States don't either. But at any rate, they don't come back for a second injection. And they said, could we develop a system that could do the following? That you could give one injection, but it will be what we'll call self-boosting. That when you look on the right-hand side, you'll give one injection, but you'll get a pulse coming out at time 0, 1 month, 2 months, maybe 6 months, a year. How could you do that? So what we thought about, Anna Jakulenik and others in our lab, is what if we made, again, little micro or nanoparticles, but we have the drug in the center, you look at the bottom, and then you have a shell, which is the gray part. If you look at this, you have this shell, and by changing the composition or thickness of the shell, we ought to make that shell be able to break at different times. And if we could do that, then maybe we could get the pulses at the different times. But this was very hard to do, so no printing process that ever existed was able to do it. So we invented a new one. Let me show you a video on that. I hope. Yeah, I heard it before, so. I'm wondering if there's something. I'll try one more time. If not, I'll try to go over it. Well, I guess somehow the video's not working. It's strange, because it was working, whoever was doing it before, it worked. But anyhow, maybe it's just me. So basically what we did is we came up, I won't go through the details, then a whole new printing approach. It's almost not only nanotechnology, but a pico technology to fill these tiny little systems. But we did it, and you could make these particles in any size. These are just some of the molds that you would have seen. But it's kind of a cool video, and you see at the end, you've made all these literally thousands of particles, but they can have different thicknesses. So we published this in Science again. We patented it. In fact, Bill Gates is one of the inventors. And the data, and this goes beyond the paper, we've continued this, has been incredibly exciting. Depending again on the compositions, which you see on the right-hand side, different compositions of polylactic glycolic acids with different molecular weights, you can control the time that these will break. And what you see is 11 different samples with 11 different timings. So the idea really is that you'd make a cocktail of these, right? You'd make a cocktail, you'd inject them in with the vaccine, and they would break at different times. So one injection really gets you 11 boosters or 10 boosters. And so that's one of the things we're working on. Just to show you one further extension of it, which we're working on again with the Gates Foundation and Mark Prousness, who's one of my graduate students, is putting them in microneedle patches. So the idea of microneedle patches, and you see a picture of microneedles on your right, is they don't go in very deep. So they really don't give you any pain, essentially. But they go deep enough so that material in them can go into the body and stay there. And so if you look on the left, what you see is a picture of one of those. It looks like a Band-Aid. And the idea, if you go into the middle, is that you just put the Band-Aid on the patient. And what we're trying to do is put these core, and we're working on this really hard, is put these core shell particles into these into these microneedles, and hopefully make them stable enough so you could ship them all over the world for patients someday. So that's one thing that we've worked on. You know, a whole other area that we've worked on, very, very different than all of the things that I mentioned so far, is not just better delivery systems, but also I've been concerned about materials and how they go into medicine. And having been at a hospital, I was curious, how did materials get into medicine? And when I looked into this, I found that almost every time it was not driven by chemical engineers or chemists. It was almost always driven by medical doctors. And what they do is they go to their house to try to find some object that resembled the organ or tissue they want to fix. So, for example, if you look at the artificial heart, that was a surgeon, at NIH saying, well, what object is like a heart and has a good flex life? And he said, well, a lady's girdle. So that's what they made the artificial heart out of. They started that in 1967. Well, now, 2022, that's still, 55 years later, that's still what it's made out of. Because once you start down that path, from an FDA standpoint, it's not easy to change. And the artificial heart, as you may know, doesn't work real well. Blood sometimes hits the surface of it, and it could form a clot. That clot goes to the patient's brain, they get a stroke, and they die. But if you think about it, something designed to be a lady's girdle, it may not be the optimal blood contacting material. And breast implants, just another one. That was actually a mattress stuffing. So we've actually tried to approach this two ways. One is designing synthetic materials from scratch, asking the question, what do you really want in a biomedical material from an engineering, chemistry, and biology standpoint, and then synthesizing. And the other was an idea that Jay Vacanti and I, a surgeon, came up with. And I thought I'd just spend just a few minutes on that. And that's the idea of what's now called tissue engineering. This is from a paper we wrote in Science. It's been cited over 8,000 times. And the idea is you could take virtually any cell type, put it on the right kind of scaffold, grow it in what we call a bioreactor, and it could make, theoretically, whatever tissue you want. Just to show you what you might do someday is, let's say you want to make, somebody comes 30 years from now, wants a new nose. And my speculation is you'll have a computer screen, and the plastic surgeon can show you 30 noses. You can have whatever nose you want. And along those lines, you could use what I'll call CAD/CAM techniques, computer-aided design, to make these noses in whatever shape you want, design them, and then use techniques like 3D printing or foaming to make them. So when you look on the left, you see a polymer nose with no cells, and it's a regular nose. But let's say somebody wanted an upturned nose. Now, it wouldn't be so hard, right? You take a little off. Let's say somebody wanted a hooked nose. I mean, they probably wouldn't, but if they did, we could do that too. And what you see on the right is taking cells, actually it could be a patient's own cells, from another part of the body like the ear and making the nose. So I thought I would just give you one real example and then mention other things you can do. And the one I'm going to tell you is FDA approved. This is skin. And here, this is a little baby that's very badly burned. But you can take the product, which are neonatal skin fibroblasts, and put them on these scaffolds and cryopreserve them. You can put them on the child at the time of the injury. This is what it looks like. Let's come back 3 weeks later, and he's doing better. And let's come back 6 months later, and you see he's pretty much healed. So these are now approved by the FDA for people who have burns and diabetic skin ulcers. Not only could you make, as I will go over, virtually any tissue or organ, you can also make these organs or tissues on a chip, which is, I think, a whole new area based on what we started with tissue engineering. And say somebody someday, the advantage of it is that it could tremendously reduce, I hope, someday animal testing and human testing. And to that end, let me just show you some work that Milika Ritzik, who's now at Toronto, and Gordana Vanyak, who's now at Columbia, both graduates of our lab. Gordana was there part of the time Maria was. And what they did, here's a heart on a chip. So you can take heart muscle, and if you put it on these scaffolds, you'll get the right way, you'll get characteristic sarcomers and intercalated discs. But what's really striking is going to be this next video that I show you, which I hope does work, because what you're going to see, if it works, is these cells beating, 6 million cells. And let's see what happens. Yeah, so this is work. By the way, we're not shaking it or anything, but it's just beating like a heart. And you can actually take what's called an EKG of this, and it looks like a regular EKG. They even started a company to do drug testing that was working with 30 large companies, had 30 different deals to look at drug testing for safety and efficacy. You can use tissue engineering to make theoretically almost anything. Skin's approved, but just to show you quickly on another slide we made, we're working on cartilage. You people may have read about ears being made for patients that don't have ears. Spinal cord repair, we're in clinical trials at one company we started. Hearing loss, another company we started. You see skin and heart. Laura Nicholson, who is also from our lab, started a company to make blood vessels, which is in phase 3 clinical trials. You can even we're working with Julie Andrews, the singer, she's helped us raise money to make new vocal cords with her surgeon, Steve Zeitels. So theoretically, again, this area is probably more in the future than it is right now, but I think it can make an enormous impact. Like I say, we're not stopping. The last thing I was going to say is, if I look at the biggest reason for the things that I've mentioned, it's really not me. It's that I've had wonderful, wonderful people, Maria and the others that I mentioned in our lab for many years. Here's a picture, every year, I hope, yeah. Oops, let me go back. I'll get there. Yeah, every year, well, before COVID, we'd always have this big lab party. I could show you what Maria looked like many, many years ago, but I won't. And this is like we have this beach house, and it's really the people in the lab that have made these things happen. And of course, the other part that sometimes people ask me is, how is my home life, and I've been very lucky. I've had a wonderful wife, and this is her, Laura. She's actually got a PhD in neuroscience, and we had 3 great kids. Here they are. Oh, I should point out, Edith Mathiowicz, who is also one of my postdocs. She's now a professor at Brown. She once did an analysis of my publications, and you could see this inflection point. So one time when they had like this reunion for me, or for the lab, but for me being old, she had this quiz. She said, well, why was this inflection point at 1986? And she asked everybody, she said, well, the answer is that's when I met my wife. So, and like I said, we've had 3 great kids. That's when they were little. And here they are a few years ago, all grown up, 2 of them actually working in biotechnology, the 3rd might too. So anyhow, it's really because of all these people that both the people in the lab and the people in my personal life that I've been lucky enough to get these things done. I just want to thank all of you again for having me here, for honoring me with this medal, and for listening to me. Thank you so much. Do I answer? I'm happy to answer questions if people want. Do you want me to answer questions? I can answer questions if people want to. It's totally up to you.
Se ofrece a... Thank you very much, Professor Lange, for this suggesting speech and for sharing with us your experience. I think that, fortunately, you don't have to look for a different job.
We'll see.
Congratulations. Si está abierto a responder alguna pregunta, algún comentario que se quiera hacer. Thank you. Yes.
Okay. Thank you again.
Thank you.
Vamos a darle la palabra al vicepresidente primero de la Xunta y consejero de Economía, Industria e Innovación para que pueda acceder al estrado.
Magnífico rector de la Universidad Santiago de Compostela, secretaria general, catedrática María José Alonso, muchas gracias por invitarnos tanto al consejero de Sanidad como a la directora general de la Agencia Gallega de Innovación y a mí mismo a estar en este acto y, por supuesto, muchas gracias a todos los asistentes. Good afternoon, everyone present, and especially welcome Professor Langer. It's a great honor to have you here in Galicia, in Santiago de Compostela. This, our city, is the end of one of the world's most important pilgrim ways. And today we're very pleased to greet you on another pilgrim. Si me permite la comparación, nuestro homenajeado seguro que también comparte la idea de que el trabajo científico guarda similitudes con el significado del verbo caminar, dirigirse a un lugar o meta, avanzar hacia él. Para el peregrino que hace el Camino de Santiago, la meta está a los pies de la catedral, en la famosa Plaza del Obradoiro, donde confluyen culturas y nacionalidades de todo el mundo, y para el científico, quizás esa meta no se vea tan lejana en la distancia, sino en el tiempo. Pero perseverar hasta llegar a ella siempre supone un pequeño paso para el hombre y un gran paso para la humanidad. Hoy la insignia que la Universidad de Santiago de Compostela entrega a Robert Langer, de algún modo es un acto de reconocimiento a su perseverancia. Todos conocemos ese poema que dice: «Caminante no hay camino, se hace camino al andar». Pues bien, no caminando, pero sí paso a paso, como nos ha trasladado en su magnífica conferencia, el profesor Langer ha abierto un camino que ha resultado ser la meta en el tratamiento de una de las mayores emergencias sanitarias que hemos vivido en los últimos tiempos, la pandemia de la COVID-19. Y si no hubiese sido por la rápida reacción de toda la comunidad científica, las consecuencias de esta crisis hubiesen sido infinitamente peores. Y eso merece todo nuestro reconocimiento y todo nuestro aplauso para las personas que, como Robert Langer, lo han hecho posible. Detrás del trabajo científico podemos decir que siempre hay un acto de generosidad, que también hoy queremos reconocer y que como Administración tenemos que saber apoyar. Sabemos perfectamente que cualquier avance en el camino de la salud es un camino largo y en ocasiones lleno de dificultades. Y después de estos años de lucha contra la pandemia, creo que como sociedad todos somos mucho más conscientes de ello y de la importancia que tiene la ciencia. Hemos vivido una crisis sanitaria sin precedentes que de algún modo ha dado mayor visibilidad al arduo trabajo de laboratorio que realizan investigadores, centros de conocimiento y empresas dedicadas a sectores como la biotecnología. Hoy es una enorme satisfacción compartir este evento no solo con maestros de esta universidad, sino también con numerosos investigadores y científicos que hoy nos acompañan. Pero cuando hablamos de la importancia de sacar la ciencia de los laboratorios, es porque el objetivo que ahora todos compartimos es conseguir que la sociedad no solo se acuerde de la ciencia cuando hablamos de enfermedades para las que aún no hay tratamiento, sino también para hacer frente a otros retos que nos empiezan a hacer una decidida apuesta por la I+D+I. Me refiero a la transición ecológica, al cambio climático o al envejecimiento activo. Y eso no es posible si no hay un trabajo conjunto del talento, del conocimiento, con la iniciativa público-privada y, por lo tanto, con el compromiso de las administraciones y con el compromiso también de la iniciativa privada, como también se ha plasmado esta misma tarde. Estamos convencidos de que otro nombre propio que hoy todos quisiéramos destacar en este acto es el de María José Alonso. y estoy convencido en este caso de que también ella lo cree así. En ella, Galicia tiene un referente hacia quien mirar. Es una profesional conocida por todos que siempre tuvo muy claro que la batalla de la COVID la ganaría la ciencia, como así fue y como ganará muchas otras. Pero María José es seguramente también el espejo en el que se miran todas aquellas estudiantes, aún, que quieren optar por una carrera científica o técnica. El ejemplo de profesionales como tú, María José, nos ayuda en el objetivo de impulsar el binomio mujer y ciencia para seguir incorporando talento femenino a la carrera investigadora. As I said before, Robert Langer has opened the path that inspires the work of the new generation of research all over the world. In our universities, in our research centers like this, in our health research institutes, in our technology centers. I finish. We advance by walking and we progress by researching. And today, Professor, as I told you before, it's truly an honor that through your person, the way of science joins to walk along the way of St. James. This advice you have received embodies all your admiration towards your work, as well as Galicia's engagement with research, development, and innovation, our commitment with the future. Thank you, Professor, for so many things. Thank you for so much.
Muchas gracias, vicepresidente. Reitero el agradecimiento inicial al Consejo de Sanidad, que se ha incorporado un poco tarde, pero ha tenido todo el interés por acompañarnos y todos lo agradecemos muchísimo y entendemos perfectamente pues, lo ocupado de las agendas, pero gracias por estar con nosotros y prestigiando este acto que es muy representativo para nuestra universidad y también al resto del personal de la Consellería y de la Dirección General de Innovación. Este acto tiene lugar, además, en un espacio simbólico, como es este Palacio de Fonseca, que albergó pues, estudios relacionados con la medicina y con la farmacia hasta tiempos recientes, farmacia hasta el año 71, y desgraciadamente tendríamos que decir que no mejoramos mucho cambiando de sitio en 1971, no fue un paso para mejor, pero bueno, estamos a tiempo de repararlo, ¿no? Pero también aquí estuvieron los estudios de medicina hasta la ubicación en su edificio en la calle San Francisco y por eso pues, es un lugar que, además de ser el edificio que representa a la universidad pues, prácticamente desde sus orígenes, pues es el lugar más noble, más representativo que tenemos y es lo que se merecía un acto como este, donde queríamos distinguir con la insignia de oro de nuestra universidad al profesor Robert Langer. Es un momento también para agradecer el esfuerzo de María José Alonso por abrir este camino, mantener esta relación académica a lo largo del tiempo, que es tan fructífera como hemos podido ver para todos, para la universidad, para nuestros investigadores y, en definitiva, para toda la humanidad. Y también pues, ha sido una satisfacción enorme el poder, desde la Universidad de Santiago, pues, lanzar esta candidatura del profesor Langer para estos premios BBVA Fronteras del Conocimiento. Ha sido realmente pues, un honor y también eso se debe al tesón, a la insistencia, a la dedicación de María José Alonso y nosotros, pues, lógicamente, lo único que podemos hacer es apoyar pues, estos proyectos, estas ideas que, como se ve, pues, son siempre del máximo interés. Muchísimas gracias por eso y por hacer todos los esfuerzos para poder tener hoy con nosotros a Robert Langer, porque yo creo que es un privilegio difícilmente pagable y difícilmente repetible. Por lo tanto, pues muchísimas gracias. Le entregamos una distinción humilde, como es la insignia de oro de la universidad, pero muy representativa porque va orientada a personas que han tenido aportaciones sobresalientes para la actividad universitaria, para el conocimiento, la investigación, la docencia. Y yo creo que ya casi la conferencia que hoy hemos escuchado merecería la insignia. Si además de eso hay todo lo que nos ha contado, toda esta trayectoria, y encima muchos hemos tenido la oportunidad de vacunarnos con Moderna, pues yo creo que, entre ellos el vicepresidente y el rector que les habla, yo creo que la insignia de oro estaba más que reconocida y acreditada, ¿no? Por tanto, pues es un acto humilde, pero que tiene que ser de muchísima gratitud por el efecto sugestivo, generador, ilusionante que personas ilustres como Robert Langer transmiten en todo lo que hacen. Yo creo que hemos tenido un ejemplo magnífico de ello. Por tanto, pues muchísimas gracias a todos por arroparnos y acompañarnos en este acto. Ha sido un placer tenerles aquí. Vamos a poder, digamos, seguir departiendo en el claustro con un refrigerio que vamos a poder tomar y también ocasión de compartir con Robert y atender también esas preguntas o comentarios que tan generosamente pues, él se ofrecía a atender después de su magnífica conferencia. Nada más, muchísimas gracias, Professor Langer. Welcome to Santiago de Compostela. Welcome to our university. We are really, really honored with your presence. The honor is for us, not for you. Thank you very much for your presence.